Hello and welcome to my video. It will be about how to input data to the Cryptogamic Russian Information System, simply CRIS. I would like to talk a little bit about structure of CRIS to begin with. So this database is united information on biodiversity of different groups of cryptogamic organisms, such as fungi, lichens, hepatic osmosis, and cyanobacteria. This source mostly contains information from herbarium, from Polar Alpine Botanical Garden Institute Herbarium of Cryptogamic Plants in Kirovsk, Russia. And also it contains information about biodiversity of these groups from the other parts of the world, but it's mostly from Russian territory. Okay, and today we going to input herbarium specimen information into the CyanPro part because we're dealing with cyanobacteria. For that, you need to go to CyanPro here or to this icon. After that, sorry, I'm gonna log out. You need to have authorization to input data to the system. By the way, this is the system work on Creative Commons license, means it's open source source of information. So we're going to log in and put username. I will put mine for now and password. Okay, here we go. We're in the system right now with authorization and with authority to input data, new data. And for that you need to go to add content. Here you will have a whole bunch of parameters and options. You can add polygon for search, you can add materials from other sources, material from literature, basic page, and other things here. And for now we will go and we will add information of this sort. This information will go to the section of KPABG. It's herbarium of Polar Alpine Botanical Garden. It's the abbreviation. So I would like to input data of existing specimen which I collected in Lapland Biosphere Reserve in 2009, and I already have some pre-investigation about that and locality. So for example, we have a lot of blank fields here, and I usually start with the latitude and longitude. In the field I took coordinates from, actually this is the beautiful place in Russia. It's Lapland State Nature Reserve. It has 4 mountains inside, and this one is a beautiful place with a lot of carbonate, calcium carbonate rock, and it's a very nice place for cyanobacteria to grow. Anyways, I collect this particular sample in this place, and I input coordinates with this specific signs. But for CRIS database, it's better to use that kind of coordinates. It doesn't matter, to be honest. And I usually copy it, and I copy it from here, I cut it and put it here. This field is very important, and it's showing you coordinate precision. I will do 100 meters. Altitude was 360. Here it's important, it's the number on your envelope when you collect the samples. It's gonna be 38109. It implies it's showing the year. And also I'm gonna check the date of sampling. I'm sorry. And it was 30th July, and I'm using this form of date European 2009. Leg name, it's the name of the person who collected sample, and it was me, Sholigin Sergey. After I switched to country, it was Russia. And usually it's pop up, the field is popping up, and you choose one. I'm gonna choose Russia. Province was hold on for a second. It was Murmansk province. And reserve, it's Lapland Reserve. And since I already had samples in the database from Lapland Reserve, I input the Lapland Reserve inside, and here I'm gonna choose Lapland Biosphere Reserve. I don't have any data about pH or light intensity, air temperature. Some of the inserators has this data, but I don't. I collected this sample from the northern ending of Manchu Tundra Mountains, southern shore of Lake Vikis, slope of the northern exposure, small water body in the Birch Elfin, and it's all this information showing the geographical location, but down the way we gonna put information about microhabitat, like shore of that little tiny lake on the sloped surface of huge massive rock, and it was under the highlight and it was wet, and we need to take it into consideration. We need to know exact micro environment of cyanobacteria, because they microorganisms, and they for them it's matter where to grow. Substrate was rock, non-calcareous rock, and determination date, it was yesterday, 26th of December 2017. And determination name, who determined them, it was again me. So right now I'm gonna show you how to input the taxa, name of taxon, to this page. You go to new species and you start. The first species was Stygonema harmoides, and I spell Stygonema, and here the first one is harmoides, and I click on that. Also, CyanoPro permits you to add some graphical information. For that, you go to Attach Media and browse. Here you choose the file, and the first one was first we're gonna upload the macrocolony, and you click upload. It shows that it's here with this icon. You click next, and here we don't have the species in the macrocolony because there is a mix of 5 species, but authority we have, it's again me, and save. And here it's showing to you macrocolony. So, but we're gonna go back to Stygonema harmoides, and we will upload a picture of that to the database. And I have it here on my desktop. Again, I should click on upload. Click next. Species, you can just do Hormoides. And authority. Next. Next species was, I believe it was Calotrix peritinum. All the same. We need to wait a little bit. Click next. And here we spell Calotrix peritinum. Authority. Means authority means it's Sergei Shelgin took these pictures and he's uploading them to the database. I'm gonna choose another one. It was Glycopsa compacta. It worked already. Compacta. And I usually use the name of species to find it easier. Compacta. And see, here we have Glycopsa compacta. The same authority. And we have the last one, which is Glycopsopsis magma. Upload. Click next. Spell magma, type it, choose it, Schilligan S, next. So, and to summarize, we have coordinates, coordinate precision, altitude, all information about your specimen. And what is important that when I will save, click to save, this herbarium specimen will automatically have the accession number in KPABG herbarium. So it's thinking a little bit and see. Actually, I messed it up a little bit, but it's good. I'm gonna show you how to edit the specimen. Here we have KPABGC, means it's portion with cyanobacteria, and 4112 is the accession number. And here you can see all data about microhabitat and habitat and who collected when and stuff like this. And also you can click on each of the pictures and you can see the macrocolony or species number 1 and species number 2. And it's obviously that in this black spot you have a mixture of algae and cyanobacteria, such as Glycopsa compacta and Stygonema harmoides. Actually, it's nice that I messed it up, and I'm gonna show you how to edit the specimen. You click edit simply, and you should be also authorized. You should log in. And here we're gonna add next one, Calotrix peritina compacta, Glycopsa compacta, and magma, Glycopsopsis magma. So right now we're done, and we have all graphical and text information about this specimen, and also we have accession number. When I'm gonna go back to Russia, and I will ship this specimen to the herbarium to which it belongs. Another thing, we need to label. Well, need to print the label, and here I'm gonna say that it's, I guess it was 4110 to 4113, and you need to press OK, and it will show you all the last specimens which been uploaded to this database, and this is mine, and I can just simply copy it and paste it to the Word file. And I prefer to make it italic to make it prettier. And I'm going actually print it out right away to put my envelope with the biomass to this envelope. And overall, after you print it, you're gonna have envelope with the affiliation. What is this herbarium? In this case, it's PUBG herbarium, Nastic commune, and you have all information about habitat and who collect that, who determined, and also accession number for this particular sample. For actually to this database can help you in the process of isolation of cyanobacteria, and for that you can create specimen scene. And if you have petri dish with some affiliation with some number, and you gonna pick up one colony from one spot and put it in another petri dish with another name, you can connect all this pages which you will create using this option, and it's much easier to keep track on what you can isolate. And also you can upload the graphical information about each petri dish. So but for huge massives of data, like 100 or even more or less, it's better to use CSV data spreadsheet or Excel spreadsheet, and our technicians, they can import this information into the database using the table spreadsheets. Thank you for watching this video. I hope it was helpful, and comment on the video, ask questions, and see you later.